Gerald Joyce here. I'm a professor at the Salk Institute in La Jolla, California. No slides, but I do have three big questions for you today, and they are, what is life? What is life in its simplest form? And can we make it in the laboratory? So what is life? Now, as you might expect, there's a lot of debate and discussion about this. So I'm just going to quote for you the so-called NASA working definition of life, 30 years old, almost 30 years old now, and I had some hand in crafting that. So let me give that to you. Life is a self-sustained chemical system capable of undergoing Darwinian evolution. I'm going to parse that for you just a little bit. Chemical. Okay, sorry, folks. You know, life in the computer, sort of Tron-like, that's not going to make it for NASA because NASA's goal is, of course, life detection on other planetary bodies and understanding the physical embodiment of life. So chemical. System, not just an individual, but a population of individuals over time and space. And now the real business part of the definition, capable of undergoing Darwinian evolution. I'm not going to explain what Darwinian evolution is. I think we know that, but let me just point to the key attributes. Self-replication, heritable information transfer across the generations, mutation, that is variation within the population, and selection. Those are the components of Darwinian evolution. And then finally, the kind of trickiest part of the definition, self-sustained. What this refers to is in an informational sense, all of the information necessary for the system to undergo Darwinian evolution is part of the system undergoing Darwinian evolution. There's no puppet master off stage. It's, you know, the play is the thing, and it's really all happening on stage. So in trying to now parse the simplest form of life, there's been a lot of attention in my lab and others on RNA, ribonucleic acid, thought by many people, I'd say most scientists, as if not the first, an early form of life on our planet. And the focus on RNA is because RNA can both carry information, genetic information, and it's a functional molecule. It can do things. It can catalyze reactions. It can build things. So think about the synthetic mRNA vaccines. Are they alive? No, despite what you might have read on social media, they can't replicate, they don't propagate, they don't evolve. They're just pure information, really. I mean, to me, the vaccines are just a delivery vehicle of information to your muscle cells to make ultimately the protein that's the vaccine. What about an RNA virus? Is an RNA virus alive? Well, it can replicate, it can evolve, but it can't evolve in a self-sustained manner. It's dependent on the host cell. Right? It's dependent on the host cell in order to evolve. But the system of an RNA virus and the host cell, you could call alive, but not the RNA virus itself. And then what we tend to focus on, which is this sort of minimal version, is an RNA enzyme, just a molecule of RNA, but it's not just the information, it's an enzyme that can make copies of itself and therefore undergo Darwinian evolution in a self-sustained manner. In principle, that could be a molecule of life. Now, how do you find a molecule like that? Well, it'd be nice if we discovered one in some weird environment, but the game is really to make it in the laboratory using evolution as the tool of invention, so-called directed evolution, to discover such an RNA molecule. And that's what we've been working on for some time. Now, when I was at SciFu back in 2010, I gave a demo of a self-replicating RNA molecule that underwent Darwinian evolution in a self-sustained manner. I didn't claim it was alive, even though it met the NASA working definition. What this was a little RNA enzyme that we evolved in the laboratory that could make more of itself by joining two pieces together. And the conjoined product is another version of itself. And actually, in a little more detail, there was a plus strand RNA enzyme that joined two pieces to make a minus strand RNA enzyme. And then that minus strand could join two pieces to make a plus strand. And it replicates like crazy. It has a doubling time of about 5 minutes. That was the demo I showed in one of the conference rooms at Google. With serial dilution, you know, grow and dilute, 10 to the 80th fold amplification in a day. We even did a so-called Google experiment, 10 to the 100th fold amplification in a little over a day. And it can evolve. There's roughly 10 bits of information in this molecule that are passed across the generations. But 10 bits is nothing, right? So the human genome has, in principle, 6 billion bits of information. A bacterial genome, in principle, has a million or a couple million bits of information. So with only 10 or 20 bits of information, our little toy system really couldn't go anywhere, limited capacity to evolve. So where are we now? Now what we're working on is a ribozyme that copies RNA letter by letter. And at present, it can copy up to about 100 letters. And it does so with an accuracy of 97% per letter. But the puppet master, the ribozyme, contains 200 letters. And so we're not there. I don't know if we can get there, but to get there, we would need a ribozyme that can copy itself 200 letters long with an accuracy of about 99% per letter. That's the game. Ever more directed evolution pressure to teach the molecule in an evolutionary sense to copy bigger things more accurately, ultimately to make its own children, molecular children. Thank you so much.